Okay. Now, this lecture is, now that we have learned what is the morphology, the shape, and the general structure of prokaryotic cells, we are going to understand more about the biology, the physiology, the biochemistry of these organisms. And so we are going to, wait, where is it? We are going to start a new chapter, which in the book is chapter 7, and that is on microbial growth. And that in the book is chapter 7, microbial growth, and here it is. So we, in this chapter, we're going to learn how prokaryotic cells grow and divide, and what are environmental factors that might interfere with the microbial growth. So we start by looking at cell division. Now, when you talk about growth in microbiology, usually when you're thinking about an organism growing with large organisms and multicellular organisms like animals and plants, when we talk about growth, often we are referring to the single organism growing and getting bigger, getting older, and all those things. This is not the case with prokaryotic cells. Prokaryotic cells are unicellular organisms with a specific size, which is fixed. And so most of the time, when microbiologists are talking about microbial growth, they are actually referring to growth of population. So growth in terms of cell numbers. You start from one cell and then you end up with millions of cells. And this is very often what is referred to as microbial growth. Now you might have, in some cases, we talk also about the single cell getting larger and big, but that is not very common. We're going to mention it when we are looking at cell division. Before cell division, the cell grows in size, it gets bigger and larger. But every time you hear in the context of microbiology growth, it is talking about the growth of a population of cells, so from a small number of cells to a large number of cells. For that to happen, cells need to divide. And how cells divide, what is the mechanism that allow for cell division? Again, in prokaryotic cells, because there is no nucleus, we cannot talk about mitosis. We talk about a different type of cell division that is called binary fission. And this is true for all the prokaryotic microorganisms. Eukaryotic microorganisms have more options when it comes to division. They can divide using mitosis in an asexual mechanism, which just creates two daughter cells that are genetically identical to the cell that started the process, or they might generate gametes through meiosis. Yeah, to make a gamete, you need to be able to reduce the number of chromosomes and go from diploid number to a haploid number. And this is impossible for prokaryotic cells that only have one chromosome. There is no pairs. Yeah, meiosis is something that can happen if you have pairs of chromosomes, so that you can separate the pairs and generate gametes. But the prokaryotic cells cannot do that. And so with eukaryotic cell, you have the option of meiosis, gametes generation, and sexual reproduction, and mitosis asexual. With prokaryotic cell, you only have binary fission. Another form of reproduction that is found in some microorganisms is budding. Now, budding is most of the time mitosis. It can happen also occasionally with the prokaryotic cell, but is basically the the term that is used to indicate a cell division where you have the replication of the DNA, the separation of the two new sets of genetic informations. But when it comes to the actual cell cytokinesis, so when it comes to the cell breaking up and generating the daughter cells, instead of having a symmetric division that generates two cells roughly of the same size, you have a asymmetric division. So there is no symmetry. One cell stays big, and you have a small portion of the daughter cell coming off. This is often seen with yeast, Khamira, when it divides. It does through budding, which is mitosis, and then this kind of asymmetric cell division. In any case, whether we are looking at asexual, binary fission, mitosis, or meiosis, before cells divide, before you have the ability to generate daughter cells or gametes, the DNA needs to be replicated, and so you need to copy the DNA. So all must replicate and segregate the genome prior to division. What do we mean by segregation is this kind of pushing the new copies of DNA to different sides of the cell so that it can be given equally to the daughter cells. For the first two lectures, I've sent today a message. If you check on ITC, there should be a message for you with links to a Google Drive link where you should find the lectures there. If you have any difficulties with getting to those lectures, do please send me a message on ITC so I can see what is the problem and how I can change it. Now, with the, sorry. There is messages I've been sending on ITC. I don't know if you guys are managing to see these messages or not. Because we started a little bit later compared to the other courses, this week and possibly next week, I'm going to add more lectures. And so I gave a lecture yesterday, and I'm giving the lecture now. And I will give a lecture on Thursday from 4 to 7 and Sunday from 4 to 7. Now with these lectures, probably we'll catch up, and you know, then we can go back to just 2 lectures a week. Okay, do you have any other questions? Are you okay? Okay, so if you don't have any other questions, I've started this chapter which deals with the microbial growth. And as I said at the beginning, microbial growth often refers to the growth in terms of numbers of cells. And so for cells to have more cells, they need to divide. And with microorganisms, we have all sorts of types of cell division. But when it comes to bacteria and archaea, because they are haploid, they only have one chromosome, they only have one option, which is binary fission. It is asexual reproduction. There is no gametes formation. There is no meiosis taking place with bacteria and archaea. In any case, cells, before they divide, they need to replicate the DNA, so they need to copy their genetic information and segregate, so pushing it in separate areas of the cytoplasm so that it can be distributed equally to the daughter cells. This schematic representation here is showing how binary fission works in prokaryotic cell. At the top here, you have a relatively small cell, which is a young prokaryotic cell. You see here, there is the cell membrane in pink, and then the cell wall. You have the ribosome, you have the chromosome, yeah? And before cell division, before binary fission, the cell grows larger in size. It makes more ribosomes, it produces more protein, and crucially, it replicates the chromosome. So you had one chromosome, now you make a copy of that chromosome, you have 2, and then you have this separation, this pushing of the chromosome on different areas of the cell, and this is done using a cytoskeletal molecule like the FTS molecule that we mentioned in when we did our first lecture. And then these 2 chromosomes are located in 2 separate parts. The cell membrane and the cell wall start growing towards the middle in vagination of these structures until you have a new cell wall that is built. This is this structure here that forms, it's called the septum, yeah? Then you have new cell wall that is built, and then you have the separation of the 2 cells. Now, this separation doesn't happen all the time. You remember, again, in the first lecture, we talked about the cells that stay attached to one another. And you can have Diplococcus, you can have Streptococcus, you can have Staphylococcus. When those arrangements take place, is because there is no complete separation of the 2 cells, and they stay attached to form maybe a chain in the case of Streptococcus. Yeah. So with bacteria, we can also define a cell cycle. Cell cycle is normally what we call the sequence of events. So what happens from one cell division to the next. So you have divided, you have a new cell. What happens to this cell until this cell divides again? As I said, bacteria divide by binary fission. And during the cell cycle, you have DNA replication and partition, and then you have cytokinesis. Cytokinesis is the word, the term that is used to describe breaking of the cell, the cytoplasm. The cytoplasm is separated and the two new cells are generated. So the first thing we said is chromosome replication. Most bacterial chromosomes are circular, so you have a circle. Because you have a circle, replication needs to, the cell, or the system needs to be able to recognize where does the chromosome start and where it ends. If the chromosome was linear, if it was, straight line, you have a beginning, you have an end, and it's fairly easy. But with the circle, unless you have something that indicates precisely where it starts, you could keep on going around in the circle and never end copying because you don't know where it begins and where it ends. So there is a sequence of nucleotide that is specific for what is called the origin of replication. It's a single origin of replication. You have a circle, and there is the origin of replication where replication begins. And there is also a point where replication ends. Now, with a circle, you might be thinking, okay, it starts here and it ends right before it starts, like I'm going in a circle. The replication goes from the beginning, from the start of replication, from the origin of replication, it goes in opposite direction. And so it does this half and this half of the circle, and eventually it ends at the terminus, which is opposite to the origin. In order to carry out the process, you need enzymes, you need proteins, you need several molecules which are involved. The DNA polymerase, of course, but there is also gyrase and topoisomerase, other proteins that are required for unfolding the DNA, uncoiling the DNA, opening the DNA, and then DNA polymerase can do the copying. And this kind of assembly or group or, multiple protein structure, if you want, is called the replisome. Yeah, so it's a group of proteins which are needed for DNA synthesis. And as I said, DNA replication goes in both directions. So it starts at the single origin of replication, and then it goes left and right to meet at the terminus. Yeah. So here there is the drawing showing, here is the origin of replication, here is the terminus. The replication, it's the replication fork, and it goes right and left until you end up with two chromosomes. You end at the terminus and you have the two chromosomes. The two chromosomes are separated, and then the cell can divide and you have two new cells. So it's a fairly straightforward and relatively simple process. Replisome, which is this group of proteins which are required for replication, is also part of the mechanism that pushes the daughter chromosomes to two opposite ends. One protein of the cytoskeleton which is involved in this process is called MREB, murine cluster B protein. You might remember we talked about this in the first lecture. It is a homolog to actin, so it's similar to actin, is part of the cytoskeleton. And it is important both for cell shape and chromosome segregation, so helping pushing the chromosome to separate areas of the cytoplasm. It is also the protein that helps with locating the origin of the chromosome, and if MREB is mutated and is not working properly, then the chromosome cannot replicate. So it is an essential protein for chromosome replication. After we've done that, there is cytokinases, and cytokinases requires squeezing the cell in the middle to separate it. Now these cells have a cell wall, so you need to the cell needs to build new cell walls. So septation is the process of building a new cell wall for separating the cells. It requires several steps. Yeah, there is first the cell needs to find where to start. Normally it's in the middle of the cell, but it's not always in the middle of the cell. So selection of the site, the place, the location for making the septum, assembly of again several proteins of the cytoskeleton that form a ring that is called the Z ring, and then linking the Z ring to the plasma membrane, and assembly of the cell wall machinery, so that all the enzymes and all this, whatever is required for building the cell wall goes there. Then you start building the cell is constricted, is squeezed a little bit, and then you have septum formation. Yeah. The Z ring formation, again, proteins that we mentioned in the cytoskeleton, like FTZ, which is a homologue of tubulin that is found in bacteria and archaea, which helps with locating the precise place in the cell where the septation needs to take place. In E. coli, there is a complex of 3 protein MIN-CDE, which makes sure that the Z ring happens in the middle, in the center of the cell. Yeah, and these work together to form to locate where the cell needs to divide. You see how here cells are dividing, yeah, and forming 2 cells. Okay, so growth. As I said, when it comes to growth, we can talk about growth of the cell itself, so the cell getting bigger, increasing cell size, or increasing cell number. And I repeat myself again, most of the time in microbiology, when we talk about growth, we are looking at growth of cell numbers. So making new cells, increasing the population, rather than growth of the individual cell. And if we look at how microorganisms behave in this process, in the laboratory, when we grow microorganisms, maybe using a Petri dish, yeah, we observe that if we put on a graph the number of living cells, yeah, number of viable cells, logarithm, because otherwise the numbers would be too big to put on a graph. Having the logarithmic system helps us keeping the numbers in a within ranges that are easily plotted on the graph. And here you have time. Yeah, so you start with one cell. Yeah, let's say, and in time, what you observe at the beginning is a first phase, a first period of time in which the number of cells doesn't change, okay? So you have the same number of cells. This is called the lag phase, yeah? Then you have a much faster growth rate here, which is called the exponential phase or logarithmic phase, where the number of cells increases very quickly, very fast. After the exponential phase, you have a stationary phase. Here again, the number of cells doesn't increase, it stays the same. And this as well can go for different times. And then after a period, you will have a death phase, whereby the number of cells now is decreasing. Yeah. So you have fewer cells than you had before. So here you have same number of cells, many more cells, same number of cells, and then less cells. And this is called the death phase. So you have these four well-defined phases and making up what's called the growth curve, which kind of summarizes the behavior of a population in culture, yeah, in culture in the laboratory, but also in nature. It's more or less the same behavior. So let's start looking at the lag phase. The lag phase, so in the lag phase, we don't have new cells that are made, but the cells that are there prepare for division. Okay, so we said 5 minutes ago, for the cell to divide, you need to replicate the DNA, you need to make more ribosome, you need to grow the cell in size. And this takes time. It might be a long time, it might be a short time. The length of the lag phase is not fixed, depending on the state of the cell. If the cell is ready for division, it might be very short. If it needs to do everything, it might take longer. Yeah. The cell is synthesizing new components. It needs to, build new material, adapt maybe to a new medium. If I am doing this in the laboratory and I'm growing the cell with one type of sugar, and then I change the sugar, maybe it needs to start producing the enzymes specific for that sugar. All these things might take place until the cell is ready to divide. When the cells are ready to divide, they start division. And from one cell you get 2, from 2 you get 4, 4, 8, 8, 16, 32, 64, and so on. And so you enter the exponential growth, yeah, the exponential phase, also called logarithmic phase, yeah. Now, during this phase, we make several observations. First of all, the rate of growth and division is constant and maximal. What does it mean? That all the cells divide at the same time, yeah? And they cannot divide any faster than this. So they have an optimal growth rate, an optimal time for cell division. E. coli might be 20 minutes if everything is perfect around them. That 20 minutes is the minimum that they need to go between 2 cell divisions. They cannot grow any faster than that. So rate of growth and division is constant and maximal. If you look at every single cell in that population, you will see that they are more or less all doing the same thing. And so the population is uniform. They are all growing, they are all copying the DNA, they are all making more ribosome, and they do it almost in synchronicity. Yeah, so the population is at the most uniform state in terms of chemical and physical properties during this phase. Yeah, and because of this, during the log phase, scientists refer to the log phase as a phase of balanced growth. Balanced growth because everybody is doing the same thing. All the cells in the population are having enough nutrients, they have enough energy, they can do the cell division with maximal growth rate. Yeah. So the cellular constituents are manufactured at constant rate relative to each other. The parts of the cell that are needed for growth, new DNA, new ribosome, new cell wall, all these things are made at constant speed. Yeah, the cell keeps on doing it without going neither slower nor faster. Yeah, and this is the logarithmic phase. Now, at some point, the number of cells grows. You have more and more cells. It will get to a point where the nutrients which are available to these cells, sugar, vitamins, iron, all those nutrients that we mentioned in the first lecture, will not be enough now to support everybody at the same time. Yeah. Besides that, you also have, because you have a high rate of metabolism, you will have metabolic products that might start causing problems for the cells. One of the very common metabolic activities of bacteria is fermentation. And during fermentation, at the end of fermentation, the cell produces acids, and acids might change the pH of the environment in which the cells are finding themselves. And so maybe now that pH is no longer as good as it was before. So after a period of logarithmic growth cannot go on forever. Yeah, at some point, the population enters a unbalanced growth. Now, unbalanced means it's no longer true that all the cells are doing everything at the same time, the same stuff at the same time all together. Some cells will still be growing fast, but other cells will be growing slower. Yeah. So the rate of synthesis is not the same within the population. Yeah. The reasons for this kind of slowing down of the growth in terms of cell numbers is very often due to a change in the availability of food, availability of nutrients, so change in nutrient level. Yeah, it could be also a change to the better. So the population was growing balanced with the nutrients that were available. If more are added, so from an environment where there is relatively low amount of nutrient, we're going to one that has more nutrients. Also, you have a moment where the cells are not all doing the same thing, and this is called shift up, or you can have a shift down from a lot of nutrient to less nutrient. Yeah. Changing environmental conditions like pH, for instance, and the production and release of waste might also cause unbalanced. After this unbalanced growth, which is the curve starts slowing down. You know, before you were in the logarithmic phase, and now you start slowing down. Numbers are still increasing, but not as fast as they were before. You enter the stationary phase. Yeah. And in the stationary phase, the number of cells doesn't change. So you have the same number of cells, total number of viable cells. You remember in that graph, it was living cells, not all cells, but only the viable ones. So the total number of viable cells remains constant, but this doesn't mean that all the cells all together at the same time have stopped dividing. You might have some that are still dividing, but some that are dying. And so the new ones replace the one that died. And so active cells stop reproducing, or reproductive rate is balanced by death rate. So I make 10 new cells and 10 have died. The total number hasn't changed. Yeah. And again, what are the reasons for the stationary phase? They are the same reason that we mentioned for the unbalanced growth. You have nutrient limitation, so there is no more food available. Limited oxygen availability for some cells that require oxygen for their metabolism. Toxic waste accumulation, like for instance, acids that might be the end product of fermentation, but also density of population. So maybe the number of cells in the space is becoming too much. So you have this critical population density. Yeah. If the environment changes in terms of nutrient ability, bacterial cells can start a program of adapting to that condition, which is called the starvation response. Yeah. So entering into stationary phase due to starvations, lack of food and other stressful condition activates this survival strategy. It's a way that the cell can try to survive and live not in ideal conditions, waiting for the environment to change and have a more, suitable environment, more food, better conditions for it to grow. This starvation strategy or this survival strategy is changing the cell, changes the cell metabolism, and changes the cell shape. Even the cell becomes smaller. It might become an endospore. You remember at the end of the first lecture, we talked about the endospore and how bacterial cells, some of them, not all of them, some of them might have the ability to change their shape and their structure to form an endospore that is resistant to environmental conditions that might be bad. You might, for cells that don't have the ability to become endospores, they might decrease in size, so get a little bit smaller, shrink, yeah, and have more condensed DNA, yeah. There are a type of protein that are stress proteins. These are called RPOs protein, which guide RNA polymerase into selecting more genes which are codifying for what are called starvation proteins. Yeah? And starvation proteins are proteins that make the cell wall a little bit harder, so they increase cross-linking in the cell wall. DPS is one of the starvation proteins which kind of tries to protect the DNA from degradation and denaturation. Chaperone proteins make sure also that proteins like important enzymes don't get denatured and damaged. Yeah. When this is happening to the cell now cannot be a regularly functioning cell. It's not a cell that is working like it was before, and the metabolism, the metabolic rate is very low. And these are surviving cells. Yeah, they're cells that are holding on, waiting for things to get better. And these are called persister cells. Yeah, cells which are entering this kind of state of survival. These are not endospores. Endospores is different. It's a different plan. With the endospore, as I said the other day, endospores can stay for thousands of years waiting. Yeah, but persister cells are okay, but they're not as strong in surviving as the endospore. But they can stay for a relatively long period of time if they are pathogenic cells. So if these are bacteria that have the ability to cause disease, and you get them in this state of trying to survive, their virulence increases, so they can cause bigger damage than they would otherwise. Yeah. And for these cells to grow again, it's not as easy. Another option is dying. Okay, so you have the cell growing fast, entering unbalanced growth, entering the stationary phase, and then when things get really bad, can become an endospore, can become a persister cell, or it might just die and that's the end of the road. The cells are old, yeah, they can become very old, the senescence, and then death phase. Now, with senescence and death phase, you have cells which might be still alive, but you cannot grow them in culture. These are called viable, but not culturable. They are alive, but not working, yeah? Or you have programmed cell death, like apoptosis. So the cell enters a program of breaking down of the protein and the DNA, and eventually dying through apoptosis. Yeah, and this is how the cell is removed from the population. Now, apoptosis in bacteria is a controlled way of death. Yeah, programmed cell death. So we understand what is the kind of behavior of a population when we grow them in culture. Yeah, when we grow them in the laboratory, we grow them on a petri dish or in a flask where the amount of nutrients is limited, the space is limited. This growth curve is not very different in the environment. It might be less regular, but is more or less the same. You have the same constraint for cells. And we said the environment changes. The environment changes all the time, and the cells need to adapt to the environment. And so now we're going to look at factors which influence growth. Yeah, so what can be in the environment, whether it's a laboratory or a natural environment, that affect growth of bacteria. Here there is a link for a nice nice video on YouTube that looks particularly at those prokaryotic cells that live in extreme environment. They are adapted for those extreme environments. So those conditions are the ideal conditions for these cells. Yeah. But most organisms live in environments which are considered moderate environments. They require concentrations of salts that are within specific range, pH, which is close to neutrality, temperatures that are neither too high nor too low. Yeah, this is the kind of environment which supports most bacterial growth. Yeah, so most organisms grow in what we consider moderate environmental conditions. But as I said before, you have others which are called extremophiles. So cells which are adapted to living in conditions that are not moderate, that require conditions that would kill other cells for growth, and these are called extremophile. Phile is liking. Yeah, extremophiles, they need to be in environments which have extremes of temperature, pH, salt, or whatever. So they grow under harsh conditions that would kill most other organisms. Here there is a table which kind of lists what are the environmental conditions which might affect microbial growth. And we're going to look at them in detail throughout the chapter, but this table kind of summarizes. You have solute and water activity. Remember osmosis. We said the other day that having a cell wall helps bacteria protecting from hypotonic environment. But hypertonic environments are difficult, and in order to grow in hypertonic environment, there is the need for specific mechanisms which make sure that there is no loss of water from the cell plasmolysis and death. Yeah. Now with the solute and water activity, we find some cells which are osmotolerant, so they can accept changes in osmosis towards the hypertonic, which they're not their ideal conditions, they're tolerating. When you tolerate something, it's you are dealing with it, but it's not your ideal status, okay? So you don't like it, but you are kind of adapting to accepting it, yeah? So these are the tolerant, osmotolerant cells. Then you have the halophiles. Halophile is not tolerating, phile is liking, yeah? So halophile, they need, in order to grow faster and grow better. The osmotolerant, they grow slower when they find themselves in conditions of high salinity. They still grow, but they don't grow as fast as normal. The halophile need high salt in order to grow faster. And the same is with pH. You have acidophile, they need low pH. Neutrophile, they like neutral pH. Alkalifile, they require pH above neutrality. Psychrophile, psychotroph, mesophile, thermophile, hyperthermophiles. These ones are cells that require different temperatures for growth. The psychrophile, they need quite low temperature. They require low temperature, below 10 degrees Celsius. Psychotrophs, they like cool temperature, not cold, cool. 0.7 might be hard for them, but ideally is below 20, yeah. Mesophile, they like middle range temperature, between 20 and 45 degrees is their range of temperature growth. Thermophile, they require high temperature. Phile, they need high temperature above 55, and the hyperthermophile, they need very high temperature. These are prokaryotic cells that live in environments like near volcanoes or near hot springs where the temperature is very high, and It's not that they're surviving those temperatures, they need that for growing. If you lower the temperature below 60, below 60 degrees, they stop growing. It's as if you put them in the fridge. So water activity, pH, temperature, oxygen concentration. Also, there are some cells that require oxygen for growth, others that are killed by oxygen. So there is aerobic and anaerobic cells, and pressure. We're going to talk about all these. We start with solute and water activity. Osmosis, as we said in the first lecture, osmosis is the movement of water caused by different concentrations of solute, and you can define environment in relation to the concentration of solute compared to the concentration of solute in the cytoplasm of the cell. And so you have a hypotonic, isotonic, and hypertonic solution. Hypotonic solutions are solutions where the concentration of solute is lower outside the cell than it is inside the cell. So water enters the cell to dilute the content of the cell down, and this causes an increase in the volume of the cell because water goes in. This increase in volume can be dangerous to the point that if the cell breaks, you call this osmolysis. However, as we said before, having the cell wall protects from this continuous growth. It limits the size, and so hypotonic environment are normally actually better environment because you get more water for cells with the cell wall. Hypertonic environments where the concentration is higher outside the cell, and so this environment take the water outside of the cell and shrink the cell are bad, generally speaking, and cause plasmolysis that is a killer of cells. Now, all cells, even our cells, have mechanisms which help them dealing with changes in osmosis. Okay, if cells were immediately killed by a small change in the concentration of solute, we couldn't have had life evolving on the planet because cells would have died very easily. So there is always a level of tolerance and and systems that are in place that kind of allow the cells to keep on working and growing and dividing, even if the environment changes around them a little bit, not too much, a little bit, but these mechanisms are in place. Extreme adaptations adds new mechanisms to the system, but in general, you can say that all cells need to be able to, have some level of tolerance with regards to their environment. You know what the water activity is? Water activity of a solution is 1/100 of the relative humidity of the solution. So it's how much water can move, basically. And how much water can move depends on how much solute is present in a solution. You remember how solute, when they are put in solution, like sodium chloride, when it's in solution, the sodium and the chloride are separated because the two ions are surrounded by a shell of water. So if you have a lot of solute, the water is occupied, it's busy keeping the solute in solution, and so you have a low low concentration of water that is free to move. Yeah. Sorry. So how much water is available for organism depend on how much solute there is. Yeah. Now, these adaptations to changes in osmotic concentration for cells that are not living in extreme environment are relatively simple systems. You know, you have a problem with solute. There is too much solute outside, or there is too much solute inside. You can change the concentration of solute inside the cell. Yeah. So reduce osmotic concentration of the cytoplasm in hypotonic solution. How does the cell know that the environment around it is hypotonic? Because water is trying to get into the cell, and when water tries to get into the cell, it pushes on the membrane of the cell, and it puts pressure on receptors, which are called or on proteins that form channels for the water movement. These are mechanosensitive channels. They are sensitive to the mechanical pressure that is caused by the water pushing to get in. Yeah. And when that happens, the cell can do something very easy, which is I make more solute inside the cytoplasm, so that I increase my concentration of solute inside, so that not so much water wants to try and get in. Yeah. And so increase the internal solute concentration using what are called compatible solutes, something that the cell needs. Yeah, something that the cell might need in the future in order to increase osmotic concentration. Yeah, in hypotonic solution. This is a mistake here. It's hypotonic solution. And so these solutes are called compatible solutes because they are solutes that something that the cell will need to use or they might need in the future, so it's not harming the cell. And this is for regular tolerance. As I said, this dealing with the environment so that you can keep on working without worrying too much about these slightly small changes in the environment. For extremely adapted microbes, there are more complex and more sophisticated mechanisms that are put in place in order to help them grow and divide in environments which have a much higher difference in concentration. So we have the halophiles. Halo from Greek means salt, yeah, and phile means liking. So you have the halophile, they like salt, they need salt for growth, they need to be in a hypertonic environment, and they normally grow in the presence of sodium chloride or other salts which have a minimum concentration of 0.2 molar, so hypertonic compared to the regular concentration of isotonic solution. Then you have extreme halophiles. These ones need a lot of salt for growth. Yeah, so 10 times more minimum, require salt concentration of 2 molar, even 6.2 molar. You know, in the Dead Sea here in Palestine, you know, Bahr al-Miyat, concentration of salt is very high. And these bacteria, there are bacteria and there are archaea there, which can grow at this very high concentration of salt. Yeah, These ones need also very high concentration of potassium, not just the sodium chloride, yeah? And almost every part of the cell is modified for dealing with the salt. So the cell wall, the protein, the plasma membrane are all adapted to this, and they need the salt. The cell has made it so that the salt is almost part of the cell structure. Yeah. Now, if we look at growth rate, so how fast the cells are growing, how fast cells are dividing, how fast the population of cells is growing, and we plot it with the sodium chloride concentration, we can distinguish 4 different types of cells. You have the non-halophile, you have the halotolerant, you have the halophile, and then you have the extreme halophile. Yeah, the non-halophiles, these guys here, in this kind of greenish bluish line, as soon as the concentration of salt goes above the isotonic point, they start dying. Yeah, they have very little ability to survive changes in tonicity. And so you see from here, which is the optimal concentration for growth, as soon as the concentration of sodium chloride increases, the number goes down and the growth rate goes down and they die. Yeah, and these are the non-halophile. Here, see, non-halophile. Then you have the halotolerant. Here, this yellow one. Halotolerant are also not happy when the concentration of salt changes. You see, from this kind of ideal, optimal concentration of salt where their growth rate was maximum, if the salt concentration increasing, the growth rate also goes down, you see? So it's similar in a way, the behavior, to the one of the non-halophile, but it's much slower, you know? You see here, the curve is going down at a much slower rate compared to the non-halophile. And so even in 2 concentration molar, 2, there is still some growth. Yeah, here there isn't, but here there is. So they can tolerate, they don't like it, they grow slower, some of them get killed, but they can tolerate it. Then we have the halophile, which are these red ones, yeah, that in these concentration that are the best concentration for the halotolerant and the non-halophile, they don't grow. There is no growth here below a minimum concentration of salt that starts making them grow faster. So increasing salt actually makes them grow faster to the optimal salt concentration, which is roughly more than 0.2 molar, yeah. And then after they get to the optimal conditions, they also start declining in their growth rate. And then you have the extreme halophiles, which are these dark blue line here. The extreme halophile, below 2 molar, they cannot grow. They need at least 2 molar salt in order to start growing, and then they start growing, and they grow faster with the increasing concentration of salt. Okay, so that is water osmosis and water activity. The other environmental conditions that might affect growth is pH. Again, you have cells, some cells which are happier at pH near neutrality, others that require lower pH, others that require higher pH. Again, you have tolerating organisms, and you have organisms that need low pH or high pH for their growth. Yeah, pH measures the relative acidity of a solution. It is the negative logarithm of the hydrogen ion concentration. Acidic pH is easier compared to alkaline pH. There is many more examples of prokaryotic cells that are happy living below pH 7. There isn't so much above pH 7. Yeah, acidophiles require low pH. Their optimum growth is between 0 and 5.5. Listen, 0, there are some cells here, ferroplasma, this is an archaea. There are ferroplasma that grow at pH almost 0, so very low pH. You have neutrophiles that like neutrality, pH between 5.5 and 7. Alkaliphiles or alkalophiles that require pH above 7. Growth between 8.5 and 11.5. There is nothing that we have been able to isolate so far that grows at pH above 12, yeah, above 11. And there isn't many environments where you have alkaline condition. Is more likely that you have acidic condition. Yeah. Again, how do the cells? What are the mechanisms that the cells can put in place in order to stop the pH from affecting them? How does pH? How does acidity or alkalinity? How do they affect the cell? They do that by having a negative impact on the structure of proteins and on the structure of DNA and RNA. So at acidic condition or alkaline condition, the proteins may denature, so they change their shape, they lose their shape, and they no longer work. And DNA also might become denatured. Also, the lipids on the membrane might have some changes in their status and structure. And so pH is important in that sense. Most microbes deal with this problem by pumping protons and keeping the pH inside the cell next to neutral. So even if they are outside in the environment, the pH is acidic and protons enter the cell can put the protons out and keep the pH inside the cell at relatively low, so neutral level. Yeah, they can do this by exchanging potassium for protons. So I put protons out, I get potassium in. Yeah, this is it's called the acidic tolerance response. Against tolerating, I need to do some work. I need to pump the protons out, but when the protons come in, actually they can bring in food. You remember, I don't know if you remember, we talked about the symporters and the antiporters, and so these protons can be used also for transporting some molecules inside the cell. Heat shock proteins, which are chaperonins, can be synthesized to avoid denaturation of proteins because of acidity. Yeah? And this issue of acidic condition or basic condition is one that microorganisms need to deal with on a almost always. And this is because microorganisms, for their nature, the way that they grow, they carry out metabolism, which produces something that is either acidic or basic. And so they do change their own pH around them. They don't need anything from outside to come and change the pH. So they need to be always working on maintaining the pH of the cytoplasm to near neutral. Temperature is another factor. They are unicellular, microorganisms are unicellular. They don't have any ways that can help them with regulating their internal temperature. And we know that proteins are easily affected by changes in temperature. They can become denatured, they change their shape, and enzymes stop working. And so high temperature can have a very negative effect on cells by blocking enzyme work, and they can, high temperature can kill the cell. If you look at any one organism on the planet, you find that they have what are called cardinal growth. Yeah, cardinal growth temperature, a minimum temperature for growth. Below this temperature, the population doesn't grow. Yeah, it doesn't automatically die. It might survive the low temperature, but it doesn't grow. Above the minimum temperature, there is a increasing growth rate, yeah, which keeps on going until you get the optimum temperature. Optimum temperature is when you have the fastest growth rate, and then above optimum, things start going wrong, and the cells start dying until you get to a maximum temperature above which the cells are dead and there is no growth. Yeah, so these three cardinal growth, temperature, minimal, maximum, and optimal, are the ones which can be defined for each bacterial cell, for each archaeal cell, easily by making some simple experiment. Yeah. Now, again, with the bacteria, because there is so many of them and they're found everywhere on the planet, you have some bacteria which grow at low temperature, some bacteria that grow at middle range temperature, some bacteria that grow at very high temperature. And so here we have the 5 different groups of bacterial cell. You have the psychrophiles. Psychro is cold, cryo, you remember cryo, maybe you remember. Psychrophiles require low temperature for growth, from 0 to 20 degrees. Above 20 degrees, they die, it's too hot. And then you have the psychrotrophs, again, kind of in the middle, cool, they like cool temperature. Their ideal temperature is in the 20, yeah, it's 0 to 35, above 35 they die. Then you have mesophile, 20 to 45, 37 is around the optimum temperature. Thermophiles, they like hotter, 55 to 85. Below 55, they don't grow. It's like you put them in the refrigerator. If you put them at 40 degrees, it's too cold, yeah? And then you have the hyperthermophiles. These ones, they need very high temperature to start growing. So above 85, 70, 80. And even though their optimal temperature usually is around the 90, 92, 93 degrees, they can tolerate higher temperature, and one species has been isolated that can survive 115, actually. This is all 115 degrees, and this is 15 degrees above water boiling. So they're very well adapted. It's not their ideal, it's not the optimal temperature, but they can still grow and divide very slowly at the temperature above 110. And so very extreme adaptation for this microorganism. Yeah. Here there is a table with some examples. It's not important. How do they adapt? How do the thermophiles adapt? Well, you have stabilization of proteins so that they don't get denatured so easily. And it could be more hydrogen bond, more prolines in the protein structure, chaperones, as we said. You have histone-like proteins stabilizing the DNA containing more G's and C's than A's and P's because it's a more stable molecule. The membrane can be stabilized by different means. You can have more saturated fats. You can have more branched and higher molecular weight lipids. For archaea, you can have the monolayers that we discussed yesterday. Ether linkages in archaeal membrane. Again, here there is a YouTube video which is nice for you to watch that kind of explains some of these adaptations for hyperthermophile. Okay. Besides the temperature, the salt, the pH, oxygen is also an important factor. Now, oxygen is a very dangerous molecule, and it is not easy to survive in an environment with oxygen. We can live in an oxygen-rich atmosphere because we have enzymes in our cells that break down reactive oxygen species, peroxidases in peroxisomes and other catalase and other enzymes that remove reactive oxygen species is what allows us to survive an environment which is rich in oxygen. But oxygen is a very dangerous molecule, and some bacteria are not able to survive the presence of oxygen, and this is because they don't have those enzymes that remove the reactive oxygen species. If the bacteria and the cells have enzymes that remove the reactive oxygen species, then they can use oxygen as final electron acceptor in an electron transport chain, and these can make them very efficient at producing energy using sugars, yeah, with the electron transport chain and the oxygen as final electron acceptor. Now, as I said, with oxygen, we have different types of cells. Again, there are the ones which are able to grow in the presence of atmospheric oxygen, which is about 20% oxygen. These are called aerobic cells. They require to be able to remove the reactive oxygen species. You have obligate aerobic cells that need oxygen for their survival. And then you have anaerobic cells. Now, with the anaerobic cells are defined as cells that can live without oxygen. Okay, so anaerobic is growth in the absence of oxygen. They don't need oxygen for growth. Now, some of them are obligate anaerobic, which cannot tolerate oxygen, that if oxygen is there, they get killed. Yeah, they get killed by oxygen. So they must be in environments that have zero oxygen. And this is because they don't have the enzymes which are necessary for removing reactive oxygen species. Yeah, reactive oxygen species are excited oxygen molecules. It could be the oxygen single, with the excited electron. It could be hydrogen peroxide. Yeah, and these are very dangerous, again, causing damage to DNA, to proteins, and to membranes, and killing cells. Yeah. Then there is the microaerophiles. Micro means small, aerophiles means like oxygen, but they like small quantity of oxygen. So they need a little less oxygen than the standard atmospheric 20%. So they grow between 2 and 10% oxygen. There are facultative anaerobes. They don't care whether oxygen is there or not. They can deal with both. If there is oxygen, they use oxygen, they grow. If there is no oxygen, they also grow. Facultative anaerobes are, not caring, but preferring oxygen. Yeah, so if oxygen is there, they grow better. And then there is the aerotolerant anaerobes, which grow with or without oxygen in a way that doesn't really affect their growth. Here we have this drawing that shows how you would find different categories of bacteria according to their relationship with oxygen. This is a test tube. In a test tube, you will have a lot of oxygen at the top and no oxygen at the bottom. So here at the top, you see here you have the oxic zone where there is oxygen, and then you have under the anoxic zone. Now, this distribution of the cells is in this figure linked with the type of enzymes which are found in these cells. So the obligate aerobe that need oxygen, so when you grow them, they will all go near the top of the test tube where they can get the oxygen. They can survive this high concentration of oxygen because they have, as enzymes, they have superoxide dismutase and catalase that both work for removal of reactive oxygen species, and so oxygen doesn't damage these cells. The facultative anaerobes also have both, and they can grow in the presence or the absence. But you see, there is a little bit more at the top because oxygen makes them grow better. Then you have aerotolerant anaerobe. They don't have catalase, this one, but they have superoxide dismutase. They don't have any preference. They are found equally distributed. Strict anaerobes need to be at the bottom. They don't have either enzyme, and so they get killed by oxygen, the cell dies. And the microaerophiles, which are like the aerobe, close to the oxygen, but not too much oxygen. These ones also have superoxide dismutase, and catalase might or might not be there. As I said, the reason for different oxygen sensitivity is the ability to remove reactive oxygen species. This is toxic oxygen that can kill the cell. You have the superoxide radical, hydrogen peroxide, hydroxyl radical, and the oxygen singlet. These ones are all dangerous for DNA, for proteins, and for membranes. Cells that are aerobic produce the protective enzymes that we mentioned before, like superoxide dismutase, catalase, peroxidase. Yeah, and these ones are the enzymes that, as soon as these products appear in the cell, as soon as the cell makes these dangerous oxygen species, they remove them, they take it off. Anaerobic microorganisms, they don't have these enzymes, or they have it in very low quantity and they cannot tolerate oxygen. They're killed by oxygen. And if you want to grow these in the laboratory, you need the special equipment so that you can make sure that there is no oxygen in the environment. Okay. Now something else which can affect growth and can cause microorganisms to die and stop growing as population is radiation. Yeah. Now radiations come in different wavelengths. You have high energy, short wavelength gamma rays and X-rays. You have long wavelength, low energy radio waves and infrared, and then in between there is the visible light, yeah, ultraviolet, infrared, and in between the visible light from short wavelength 400 violet to the 760 roughly of red wavelength. Now, visible light is relatively low energy, it doesn't cause damage. The high energy gamma and X-rays can cause very serious damage and even ultraviolet to DNA. Now, light can also cause some damage, but it has to be very intense and for a long period of time. So radiation damage, ionizing radiations, X-rays and gamma rays, these are penetrating. They have very high energy, low wavelength, high energy, so they can enter, they can penetrate a cell wall and the cell and cause damage directly to the DNA. They cause mutations, which can lead to the death of the microorganism. We can use these X-rays and gamma rays to clean and sterilize objects, like plastic objects that cannot be boiled because they will melt, like Petri dishes or syringes, you know, any of these materials that need to be cleaned but that are delicate at the same time can be sterilized using X-rays and gamma rays. And they disrupt chemical structure of many molecules, including DNA. Some bacteria also are adapted to dealing with this problem. Now, if you are a bacterium that lives in a place which is exposed to the sun for a long time, like if you live in the Dead Sea, again, you know, in the Bahr el-Miyat, there isn't trees, there is nothing that can protect you. You are exposed to the sun, and the sun can, you know, produce a lot of ultraviolet rays. You need to be able to protect yourself from this danger. And so they have enzymes which fix and, repair any DNA damage that might be caused by this radiation. Now, they can do it again to a certain extent. They cannot do it for a very extensive damage, but the small damage, small dose, they can actually repair and fix this problem. There is one species of bacteria, Deinococcus radiodurans. This is a very famous bacterium because it can survive very, very high level of very high dose of ionizing radiation that causes the DNA damage, and it can keep on fixing and repairing the DNA. And so this is extremely resistant to ionizing radiation. Yeah. Ultraviolet radiation causes mutation by formation of thymine dimers in DNA. It is a dangerous radiation, but it's not as penetrating as the ionizing radiations. It can be blocked by surfaces. Yeah, so if it is like if the microorganisms are below a bubble of water, the UV cannot penetrate. If there is plastic covering, the UV cannot penetrate. So it needs direct exposure on the surface of the microbes. Yeah. And again, for thymine dimers, you have repair mechanisms which are possible. As I said, visible light also can cause some damage. It has to be high intensity of light, but at high intensity of light, again, you have the singlet oxygen, which is a powerful oxidizing agent. This could be a problem for organisms that are photosynthetic, because if you carry out photosynthesis, you want a lot of light, so you are exposing yourself as much as you can to light. But if there is too much light, that can damage. And so pigmentation helps. Pigments like carotenoids and other accessory pigments can help by absorbing a little bit of this light and making it less damaging. Yeah. So this process of damage caused by too much light intensity is called photooxidation. Okay? Let me see how many slides do we have. Okay, there is only 12 slides left. So let's finish this chapter, microbial growth in natural environment. All this that we discussed was discussed as if something that is done in the laboratory. I can measure how much do they need. I can measure how much salt, I can measure temperature, the oxygen and all that. In natural environment, in outside of controlled conditions, we can look at how microorganisms grow. As I said, they follow more or less the same growth curve, but there are some variations. Yeah, you need to take into consideration that always microbial environment are not simple, they're complex. They change all the time. They often have very low amount of nutrients. They're called oligotrophic environment. And they can also be the places where dangerous conditions, which might kill the organism, are present. Yeah, and so microorganisms need to be flexible in their response to natural environment, and need to be able to respond to this kind of challenging condition. One way that microorganisms have to kind of deal with these conditions that might be challenging is by making biofilms, okay? Biofilms are structures that are made by several microorganisms that are living together, microorganisms of different species, and different genera might grow together, attached to surfaces, so they stick to the surface of something instead of being free floating around. Now these surfaces can be inorganic surfaces, like a rock in a river, or a hull of a boat in the sea. You know, sometimes in the sea you can see these lines, greenish lines, where the water meets with the air on the boat, and these lines is biofilms of microorganisms, but they can grow also on organic material. They grow on our teeth, the kells, the plaque on the teeth is the microorganism. They can unfortunately grow also on implants, surgical implants, like hip replacement, or they can grow on catheters, and so this is a choice that microorganisms have. They live in a community, they form a biofilm. They are very useful because they can protect microorganisms from the environment, and with communities you can exchange, and you can have products of metabolism of one species being used by another species, and so when you look at a at biofilm, you find that there is a matrix in which the organisms grow. This matrix is made mostly of proteins, sugars. There is even some DNA in it. It's the extracellular polymer, or matrix. It is usually has a negative charge at the top, and so antimicrobial agents that might be positively charged cannot enter. So it protects from antibodies, it protects from antibiotics, it protects from toxic material. It also generates different gradients of oxygen. In the middle, there is very little oxygen, so you'll have anaerobes growing here. In the near the surface, there is more oxygen. You can have aerobic cells here. Cells talk to each other through signals, and they can change their behavior depending to the signals that they receive from other cells. They can exchange DNA, they can exchange plasmid, they can exchange genes that make them better. So biofilm is a very nice kind of environment for cells to grow in. It forms by cells sticking to surfaces. The surface needs to be conditioned by the presence of something organic, proteins, glycoproteins, sugars, they might be there, and then they start, when they're there, they kind of stick there. This first attachment is not very strong. They can come off and go back on, but then when they start producing this exopolymer, when they start producing the matrix, and they start signaling to each other and talking to each other and deciding to build this community, then the biofilm becomes something which is very strongly attached and very difficult to remove. They start secreting polysaccharides, protein, DNA, and then you have this growth of the biofilm. The mature biofilm is complex, dynamic community of microorganisms. It contains different species, is heterogeneous, have different species, different metabolic requirements, different metabolic activities, different species there. And you have interactions, exchange, DNA, uptake, communication, resistance to danger. Yeah, all this is possible. The extracellular polymer, which is this EPS, which is this extracellular matrix, protects the microbes from UV light, antibiotics, antimicrobials, toxins, anything. And if this biofilm form on medical devices like catheters, implants, and so on, that can cause very serious illness. Yeah. Now these microorganisms can also start growing a biofilm on structures like water tanks. Yeah, the water tanks that store water, if the water tanks are full all the time, then you don't have biofilm. But if they are half empty, you have air at the top and the water at the point of where the water is, finishing. There you can have biofilms, and in these biofilms you might have microorganisms that can cause disease. And so sometimes when these form in water storage facilities, if the microorganism starts coming off and entering the water, then they can contaminate drinking water. Yeah. We said that cells in biofilms talk to each other, they communicate using signaling. And the signaling, one of the ways that microorganisms have to communicate with one another is by using quorum sensing, which is a system depending on the density. So how many cells of the same species are present? If I have cells of the same species, I want to be able to communicate with them because we can help each other. Yeah. And this is called quorum sensing. Quorum sensing is based on signals and receptors. So the cells produce signals, which are small proteins, which are released. And if there is many cells of the same species, the concentration of these proteins will be quite high. And then they can be received by a receptor that is found on the membrane of another cell, or the same cell actually can self-stimulate. And when they do that, they change the behavior, the status of the microbe, and the microbe becomes what we say competent. Competent, what does it mean, competent? Competent means to be able to do something, yeah? And in this case is to be able to accept things like DNA. They can pick up DNA, they can release antimicrobial agents that might kill bacteria of a different species, yeah? So they kind of protect each other, and this mechanism was discovered in gram-negative cells first, in Pseudomonas aeruginosa. Acylhomoserine lactone is a molecule produced by many gram-negative organisms. This is the auto-inducer because it can be released and go to signal to cells next to the cell that produces it, but it can also be received by the same cell that produces it. And then once it is received by the signal, it causes the change behavior like expression of genes, which can regulate the different functions. And here you have in this table different examples of acylhomoserine lactones and other other signaling molecules. Where are they found and what is the function that is regulated? We don't need to worry about this too much, but okay, so we're almost at the end of this chapter, and the last thing that we need to discuss is, okay, we understand all this. We understand how environments regulate cell growth. We understand how the cells grow, lag phase, logarithmic phase, stationary phase, death phase. You know, thank God for that. Otherwise, we will be swimming in bacteria if there wasn't that. But sometimes we want to keep bacteria growing. We don't want them to die. We don't want them to enter the, you know, the death phase. This is when we are growing the bacteria in order to use the bacteria for some product that we want. Antibiotics, we might want enzymes for making soaps, for washing clothes, whatever, vitamins, yeah? So when we are growing bacteria in order to extract something that is useful for us, we don't want to have this growth. We want them to be always growing, and most of the time we want them to grow in a logarithmic phase, but sometimes we want them just before the stationary phase. For instance, antibiotic production is better near the stationary phase. Yeah. So when we want to do that, we don't want to have the bacteria growing in a closed system, like a petri dish or a flask with broth in it. We want them to grow in an open system where I can keep on changing the conditions in order to keep them in that optimal range, which makes my bacteria work for me. So this is an open system. They're called the fermenters. Yeah? You continually provide nutrients. So the bacteria eat the food, they consume it, you add some more, and you remove the waste that might cause slowing down of growth rate. So you keep the cell in a constant phase of growth. Most of the time is log phase, but it's not always log phase. Yeah. So we constantly supply cells in exponential phase and keep the conditions in the optimal, in the optimums, where you have the maximum growth rate. You can use this for studying growth. You can use this to study what is limiting nutrients for different organisms. But most of the time, this is something that is done for food and industrial microbiology in order to produce something that is useful. When I want to make yogurt, for instance, I need to have microorganisms. I need them not to die and to stay there. Yeah, the open system of, why is it going like this? The open system, which is used for this, is called the chemostat. Yeah, in the chemostat, this is, you see here is the place where the bacteria are growing. This is the culture vessel, the place where the bacteria are growing, dividing, and producing whatever I want to take from them. I have fresh medium containing nutrients, which I can add. Yeah, so this is a control valve that allows me to put fresh food, fresh medium, and so on. There might be air supply if it is aerobic culture, but I need also, you know, as much as I add, I might have to take out because I want to remove the waste. I don't want to increase the volume. I don't want to dilute the culture. I want to keep it always in the same condition. Yeah. Usually, in order to in order to regulate the growth. An essential nutrient like a vitamin or an amino acid that the cell cannot produce themselves is kept, it is the one that I measure the concentration so that I keep the cells in the phase that I want. Yeah. Okay, so this finishes this chapter. So we're doing okay. We finished 3 chapters, and I will send the link for this. Now the link for this is going to be a Zoom link. Before I had the setting wrong in my computer, and I wasn't recording the lecture on the cloud. I was recording it on my computer, and that is why, you know, there were some little problems with giving you the link for those lectures, but this is now recorded on the cloud, and so the link is a Zoom link, which I will send as, you know, before tomorrow. Tomorrow there is no lecture, but on Thursday we are going to have a lecture at 4 o'clock. Okay, yalla, good luck.